Let us consider another class of promoters in which RNA polymerase binds efficiently and unaided and forms a stable closed complex. But that closed complex doesn't spontaneously undergo transition to the open complex. At this promoter, an activator must stimulate the transition from a closed to open complex, since that transition is the rate limiting step. तो यहाँ पे transition is the rate limiting step. Activators that stimulate this kind of promoter work by triggering a conformational change in either RNA polymerase or DNA. So they cause a conformational change in the RNA polymerase or DNA. It means that they interact with the stable closed complex and induce a conformational change that causes transition to the open complex. This mechanism is an example of allostery. So here you can see this is activator binding site, and when the activator binds, it causes some conformational change. Here is the conformational change that takes place, and it leads to the transcription. For example, the GLNA promoter is the example of such, and NTRC is the activator, which is example of such a case. In still another class of promoter, transcription initiation is limited at the step of promoter escape. One example of such a promoter directs expression of the malT gene. This is an example of such a case. In the absence of an activator, it undergoes abortive initiation, and only in the presence of an activator will it efficiently escape into elongation. Repressors can work in ways other than just blocking the binding of RNA polymerase. For example, some repressors interact with polymerase at the promoter and inhibit transition to the open complex or promoter escape. Generally, DNA binding proteins that interact with each other bind to adjacent sites, but some proteins interact with each other even when bound to sites well separated on the DNA. To accommodate this interaction, the DNA between the sites loops out, bringing the sites into proximity with one another. तो जैसे मैंने कहा जी कि ये डिस्टेंस पे भी हो सकती हैं ये जो प्रोटीन्स हैं, तो जब ये कुछ सफिशिएंट डिस्टेंस पे होगी, इस केस में ये जो इन दोनों के दरमियान में जो डीएनए है दैट विल लूप आउट, और इस तरह से इन दोनों की बाइंडिंग हो सकेगी. डिस्टेंट डीएनए साइट्स कैन बी ब्रॉट टुगेदर or closer to help loop formation by this loop formation. In bacteria, for example, there are cases in which a protein binds between an activator binding site and the promoter and helps the activator interact with polymerase by bending the DNA in a favorable direction. There are also cases where such a protein hinders loop formation and activation by bending the DNA in an unfavorable direction.